So hello everyone and welcome to the third installment of our Neurophotonics Neuro Light Lunch. Thanks for joining us. Today we are really lucky to have two speakers, both hailing from Janelia farm. Our first speaker is going to be Kaspar Podgorski, who is developing hardware and software and algorithms for image analysis, especially in vivo imaging. Our second speaker is Ahmed Abdelfattah, who is also he's a postdoc at Janelia, and he is developing chemogenetic probes for imaging brain activity, many of which you'll all be familiar with. They're really, they've made a big splash in recent years. So I'm not going to take a lot of time because I know you want to hear the speakers. So I'll just start by very quickly introducing Kaspar. Kaspar grew up in Toronto. He actually did his PhD studies in the lab of my friend and colleague, Kurt Haas at UBC, and that's where I first met Kaspar. We were lucky enough to have some collaborations and interactions back then, and that's where I got to know him as a really dynamic and brilliant guy, kind of force of nature, although it's fair to say that sometimes nature kicks his butt, but we're lucky that he's here to join us today. Kaspar has been at Janelia Research Campus, where his lab is, as I said, developing hardware, algorithms, and indicators for imaging neural activity in vivo. And I'm happy to say that in 2021, he'll actually be coming back to Canada for a position in Toronto at the University of Toronto and Sickkids. So Kaspar, I'll hand over the podium to you.
Uh-oh. Okay. You cut out for a second there, but I hope you can, everyone can see me, and everyone can see my screen.
Oh, before you, I just want to say before you,
oh, sorry,
that for questions, will you please put your questions into the chat. That seems to have worked well last time. And then at the end of the talk, we'll go through the questions one by one. People can ask their questions in person, but if you put them in the chat, we'll know you have a question. Thanks.
Great. Thanks very much, Ed. When I submitted the title for this talk, I promised to talk about some new DMD-based microscopes, and I will touch on those towards the end of the talk. If you're interested, ask me afterwards, because I sort of forgot and I didn't put that many slides on that in here. But everyone, my name is Kaspar Podgorski, and I'm going to talk more generally about my lab's work on speeding up two-photon brain imaging. Of course, a big part of neuroscience has been using microscopes to record the activity of neurons and other brain cells. And speed has been really important for these types of experiments. So the video that you're watching right now is calcium imaging in a mouse's hippocampus in an area called CA1 using a large field of view raster scanning two-photon mesoscope. And so in CA1, these cells, many of these cells are place cells. They encode an animal's location. And a really interesting phenomenon is that during rest, those neurons replay past experiences. So the place cells will fire in the same order that they did when the animal was moving around. And that all happens at super high speed for reasons that nobody really understands. So those replay patterns, they actually occur on timescales of milliseconds in neurons that are scattered across many millimeters of tissue. So they're too fast for a raster scanning microscope like this one to capture. And if replay events are occurring in these videos, they're just looking like near simultaneous flashes. So this is one case where it's just really obvious that we need faster microscopes and we need optical indicators that can follow activity on millisecond timescales. But you don't have to image large volumes to run into the speed limits of existing microscopes. So each of those CA1 neurons performs computations, like producing its place field, by combining thousands of synaptic inputs. And we have no idea how that happens, because even though it's possible for us to record synaptic activity, for example, with calcium indicators, we don't have microscopes that can record from enough inputs at a time to study how neurons compute. So we really don't know what patterns of input come into these cells or how they're transformed to produce the fascinating output that those neurons produce. Okay, and final motivation, maybe it's a really obvious one, is that some signals are just really fast. So neuronal calcium signals are fundamentally slow, which is actually a good thing because it makes it possible to record them with traditional existing slower imaging methods. But it makes it hard to, for example, count or measure the precise timing of action potentials. Calcium indicators also don't detect subthreshold input, like the depolarization marked by the white arrows here. So there's been a lot of recent development of indicators for these sorts of subthreshold signals, and a lot of those indicators are really, really fast. So you'll hear about voltage imaging from Ahmed, who developed Voltron. But here is another example of a voltage indicator. This is ASAP3 from Michael Lin's group. And these voltage indicators have decay kinetics on the order of a couple of milliseconds or so. Lauren Luger's lab has produced fast sensors for neurotransmitters that are sensitive and specific. Glue sniffer is perhaps the most popular. It detects glutamate, the main excitatory neurotransmitter in vertebrates, and it has a time constant of about 10 milliseconds in vivo. GABA sniffer, which detects the main inhibitory neurotransmitter, has a time constant of tens of milliseconds, and acetylcholine sniffer detects in vivo release with time constants of a few milliseconds. So what all these indicators have in common is that they're much faster than calcium signals, and we need faster microscopes to record them. So all this is to say that there are many different applications that desperately need faster two-photon imaging techniques. And whether that's to record from larger volumes with conventional indicators, to do intricate recordings of smaller volumes, like measuring synaptic input patterns to single neurons, or recording new indicators and neural signals that simply require higher bandwidths. So there's a wide community of optical methods people, and many of you are here, that are out to address these challenges using a variety of new techniques. So the traditional approach to two-photon imaging is to create a point focus and then raster scan it pixel by pixel through a volume of interest. Under that approach, you get a faster frame rate by speeding up your scanners to move the focus faster through the volume. And a lot of interesting work has gone into doing just that in various ways. But there's a fundamental limit to how fast raster scanning can ever go. And we already have scanning methods that are hitting that limit. And that limit is set by how long it takes for light to come out of the sample, which is on the order of 10 nanoseconds. Fluorophores just live in an excited state for a certain amount of time before they decay. So we can make at most about 10 to the 8 independent measurements per second. And that means that the fastest that you can ever record a 1 megapixel image is on the order of about 100 hertz. So it's clear that if we want to image faster, we have to make our measurements more efficient. For recording neural activity, traditional raster imaging is extremely inefficient. So a typical field of view might contain a few hundred neurons that we care about, for which we only want the total brightness of each of them on each frame. So it's a few hundred numbers. But we often record millions of pixels per frame to produce those few hundred measurements. So several ways have been explored to make two-photon measurements more efficient. And the optical techniques fall generally into two categories: random access imaging and projection microscopy. So the first is random access imaging. And instead of scanning through every point in a volume, random access imaging uses optics that can jump from point to point. But there's a cost in making those jumps, and that's around 20 microseconds or so usually. But if you know the points that you want to image in advance, there aren't too many of them, and they're not moving too much, importantly, then you can save a lot of time by not scanning all the intervening space. So this is something that I actually did during my PhD. It's an approach that works really well when you don't have a lot of sample motion, and it works great in tadpoles. So in Kurt Haas's lab at UBC, I built one of these microscopes for imaging whole neurons in the developing tadpole tectum. And that allowed us to study how neurons sort of compare local calcium signals to global spikes to direct their growth. Importantly, these are very small neurons with only about 400 synapses. And again, the tadpoles can be held immobile. But there's also another family of efficient imaging approaches that I'm going to call projection microscopy, because they deliberately combine multiple voxels into each measurement. And you'll see that the sort of mathematics that govern these approaches can be quite different. So an example of projection microscopy is Bessel beam excitation. So here, instead of making a focus that looks like a point, you make one that looks like a pencil elongated along the z-axis, or z-axis, sorry, I forgot this in Canada. So when you scan a Bessel beam in 2 dimensions, you collect an axial projection of a 3D volume. So it's basically the sum intensity of your sample along the z-axis. If your sample is sparse enough, then you can get all the information you need out of the volume in just one frame without axial scanning. Another approach is to produce multiple distinct foci and then scan all of them together. The image that produces is the sum of the images that would be produced by each of those individual foci. So again, if information is sparse enough in your sample, you can analyze those sum images directly. And if your signals end up overlapping, you can use computational unmixing to recover them. So these two approaches each have advantages and disadvantages that can complement each other. And importantly, they're not mutually exclusive. So we thought, why not do both? Like they could be combined to trade off their strengths and weaknesses. So we built a microscope that uses parallel excitation to speed up imaging from targeted sample regions. So we started by developing a tomographic two-photon microscope. Instead of illuminating a point, it illuminates a whole line at a time. It scans that line of excitation across the sample plane at multiple angles. So then at any given point in time, the measurement that it makes is the sum of the pixels along the line. The resulting measurements are angular projections of the 2D sample plane, a lot like the measurements in CT imaging. But unlike Bessel beams, these excitation lines lie horizontally within the 2D focal plane. And in this microscope, we're collecting 4 angles spaced from each other by 45 degrees. There's a refinement to this, which is that we add a spatial light modulator that acts as an amplitude mask. It blocks most of the excitation light from making it to the sample, and it only lets light through in the regions that we want to measure. So this is really important. It reduces background excitation, and it reduces the size of the focus at any given time, which makes two-photon excitation more power efficient. So we call this technique scanned line angular projection microscopy, or SLAP for short. So the designs for this microscope are publicly available. The microscope, including the laser, can be built for around $320,000. So SLAP has a unique combination of strengths. It matches the resolution of conventional two-photon imaging along the scan axis. It's very fast because it records from the entire field of view in just 4 line scans. So we routinely image at over 1 kilohertz. It inherits the optical sectioning benefits of two-photon excitation. It's robust to sample motion because we can record a little buffer area around each region of interest. And then we can register out motion post hoc like you do in area imaging. It allows for accurate mixing because the line measurements form what's called a low coherence basis. This is essentially saying that 2 lines only ever cross each other at a single point. So measurements tend not to mix multiple pixels together in the same way, and it makes it easier to recover the sample from the measurements. And because the SLM blocks most of the excitation light, SLAP is more power efficient than other line scanning methods. And we typically use about 100 milliwatts to image in vivo at 1,000 hertz. So let's talk about how you transform SLAP measurements into images. The tomographic basis allows us to make measurements that depend on every pixel in the image without having to make a separate measurement for every single pixel. So SLAP performs about 5,000 measurements per plane, and that's not enough to recover an arbitrary megapixel image. But real images have a lot of structure. So by using priors that define what kinds of images are possible, we can reconstruct the pixel space images from relatively few SLAP measurements. So the use of priors like this has been well studied in medical imaging, and similar approaches have been used with MRI and CT. And as I'll show you, the priors that we can use for activity imaging are much, much more powerful than what's been done in CT or MRI. The best introduction to this type of compressive tomography is particle localization. So let's go through that. So let's say you have an arrangement of fluorescent particles in your focal plane. So on the left here is an image of fluorescent beads on a glass slide. As each line scans the sample, when that line crosses one of these dots, these fluorescent beads, it produces a bump of fluorescence at the corresponding location along that axis. The locations of the bumps correspond to the coordinates of the particles along the 4 line axes. So on the very right is what's called the back projection image. It's made by adding a picture of the line at the point where it was every time a photon was detected. And in the back projection, at every place where there's a particle, all 4 lines intersect. But the back projection isn't how you perform reconstruction. So we perform reconstruction, maximum likelihood reconstruction, using Richardson-Lucey deconvolution. And that's a simple iterative algorithm that can start with the back projection, and it converges towards the solution image. For relatively small numbers of particles per plane, we recover their locations without errors at extremely high speed, over 1 kilohertz, and high spatial precision. To give you a sense of this microscope speed, this is a movie of particles flowing through a 3D imaging volume. The volume consists of 100 planes, and we record 10 volumes every second. Every plane contains about 1.5 million pixels and takes less than 1 millisecond to collect. So the recording is about 1.5 billion voxels per second, and that's 15 times the theoretical limit on raster scanning. And the way we broke that limit is with compression. So SLAP is really fast because it collects images directly in a compressed representation. And an interesting finding is that you can decompress that data very well as long as you know some compression scheme for the original images, as long as you have a model, basically, that describes a restricted image space. So for particle tracking, like in the video that I showed you, that compression scheme can be the locations of the particles, which is a very efficient representation. For activity imaging, we have a very different representation, which is also very efficient. And in that, we take a static picture of the sample in advance, and we carve it into segments. So this is what it looks like. We start with a high-res image stack, which we acquire in a way that avoids motion artifacts. And then we segment that into up to about 1,000 segments per plane. The segmentation is done with a pixel classifier that we train using supervised machine learning, and it's designed to identify individual dendritic spines. But you can use any segmentation you want as long as the number of segments is small enough. The underlying assumption here is that the activity within each of these little segments is homogeneous. We then solve a model to recover the segment activity over time. And that model looks like this. We make measurements y, which are Poisson, because we measure integer numbers of photons. And they come in at a rate that's the product of the projection matrix P, which describes how each measurement samples voxel space, times the segmentation, which groups voxels into segments, times the activity of each of those segments over time, plus a baseline. We also model the dynamics of the indicator, which rises quickly as spikes and then decay exponentially. So the only thing we need to estimate here is the spikes for each segment. The biggest challenge is knowing the projection matrix, P, which describes how each measurement samples space. So this requires a very precise calibration of your microscope. And we include things like the imaging distortion and the difference between the raster PSF and the line excitation PSF. You can know if that model is right by comparing your expectations to measurements. So the blue line is the expected data using our measurement model given the reference image. It's mathematically projecting the reference image with our measurement model. The orange line is the actual measurements that we get. And there's a very good agreement, which means that the model is good. So we did several experiments in vitro to validate this method in conditions where we know the ground truth activity. So one of the first things that we did was uncage glutamate at 2 locations on a glue sniffer expressing neuron spaced by a gap of 10 milliseconds. So this movie was recorded at 1,016 hertz and is a single trial. So we're showing delta F over F, which is the change in brightness of the indicator, as the redness of the pixels. The purpose of these experiments is to get a sense of how well we can observe patterns in time and space. So if we plot the measured brightness of the 2 uncaging locations, we can recover the 10 millisecond delay that we put into the experiment. We can also watch diffusion happening along a segment of dendrite and recover the known diffusion coefficient of the small molecule glutamate in solution. We've also imaged voltage indicators, showing that we can record spikes from single neurons with high signal to noise, and even small clusters of neurons. But where two-photon microscopy really shines is for in vivo imaging. So this is a raster scanning movie of glue sniffer, where we're showing a mouse visual motion stimuli. And you can see lots of activity when we show the stimuli in both dendritic shafts and spines. So these delta F over F responses that you're looking at have been conditioned on Z-score with a high threshold. So all the red signals that you see are actually quite large, but they're hard to appreciate because most of the flashes are only there for a single frame, since glutamate release is so fast. So this movie is about 3.4 hertz. A resonance scanning microscope could record the same field of view at 15 hertz, but that would still be way too slow for this indicator. And mostly what you can tell from these raster recordings is that we're missing a lot of the action. So this is a slap movie of the same field of view over the course of a single stimulus presentation. At t equals 0, the stimulus starts, but it takes about 200 milliseconds for activity to reach the cortex. At that time, a few spines spike before the others. As the stimulus continues, a diffuse glow starts to appear, showing you where the local neuropil is most active, as individual spines also continue to spike. Some dendritic shafts also show clear spikes, and so these can correspond to an adjacent synapse belonging to another neuron, or a spine pointing out of the image plane, or even a synapse that doesn't come onto a spine. And it takes a little over 1 second for the stimulus to complete one cycle. At this time, some of the spines start to repeat the pattern of activity that they showed during the first cycle. And that means that those spines are sensitive to specific phases of the grading as it moves across the screen. And so a little over 2 seconds after it started, the stimulus is going to stop, but then again, it will take another few hundred milliseconds for the activity to die down. So we can compare tuning maps made from raster scanning to SLAP recordings, and they agree very well. In fact, for a fixed number of trials, the SLAP recordings are better at predicting the raster tuning than raster recordings are themselves. And so, for example, this is the activity of a single spine over 64 trials for 8 each of the 8 different motion directions. And the black lines here denote the start and end of the stimulus period. And from recordings like this, we can ask what's consistent, what's variable about the timing of evoked activity, and how responses evolve over millisecond time scales. So for SLAP imaging to work, samples need to be sparse in some sense. They can either be sparsely labeled, like the dendritic imaging that I showed you, or the sample can be densely labeled, but only be active at a few sites at a time. Acetylcholine release is much sparser in cortex than glutamate, and we wanted to see if we could densely label visual cortex with acetylcholine sniffer and use whole field SLAP imaging to identify sites of acetylcholine release triggered by electrical stimulation of the basal forebrain. So even though the label is dense, if we look at the intensity on each of the 4 scans over time, you can see these brief localized blips of fluorescence following stimulation. These release events have a spatial extent of about a couple of microns and last about 10 milliseconds. So we'd never be able to find them with traditional imaging. After we localize each of these events on the 4 different axes, we can use back projection to find their precise location in 2D space. And those lines always intersect, and they identify the point where that release occurred. And then we can use the SLM to generate more targeted patterns for subsequent imaging. So this wide field scanning approach works. I'd say the main limitation is the laser power involved, which limits these wide field scans to a couple of seconds at a time for a larger field of view. I also want to mention other projects to give you a bigger picture of what my lab does. So my lab essentially works on methods for studying computation in single neurons. And to make that possible, we're building microscopes like what I've shown you. We're evolving genetically encoded indicators and developing algorithms. And we're also using these tools to study how cortical neurons turn inputs into outputs in mice. So my lab's going to be moving to Toronto in 2021, as I mentioned, and I will have several positions available there. So as I talk about these projects, if any of them pique your interest, please don't hesitate to contact me. I'd really like to hear from you. So in microscopes, a really big effort has been developing SLAP2, which is a microscope that at several other labs are also cloning. So this is a DMD-based two-photon microscope that's similar to SLAP in a lot of ways, but it's faster, it's easier to use, and it shows you images in real time. So it has 2 fields of view that can be independently targeted in 3 dimensions. And the speed of SLAP2 depends on how many objects you image. But it can record activity from thousands of objects, which might be synapses or they might be neurons, at over 1 kilohertz each without any computational post-processing. So you see the results instantly. It can also do tomography at 20 kilohertz using post-processing similar to what happens with SLAP1. SLAP2 can also do very rich optogenetics with thousands of targets per pattern and a pattern rate of 20 kilohertz. So in collaboration with Ahmed, as well as Eric Schreiter, Lauren Luger, and the Genie project, we've been improving indicators for two-photon imaging. A big part of this has been making indicators compatible with inexpensive and powerful 1030 nanometer lasers. So these lasers would make two-photon imaging much, much cheaper and easier, but green indicators aren't great at this wavelength. So we've been converting high-performance green indicators into yellow ones. So I already told you about our work with glutamate and acetylcholine indicators, both of which were yellow versions that Lauren Luger's lab made in collaboration with us. But we also recently, my lab made JY-Camp, a yellow version of G-Camp. So this here is an orientation tuning map comparing G-Camp 7 to JY-Camp on the same scale under 1030 nanometer excitation. And the indicator is available on AdGene, so please give it a try. It's especially good for 2-color imaging along with a red calcium indicator. Another really important project has been improving glue sniffer, and that's been led by Adi Agarwal in my lab. So he has a new glue sniffer 3, which is darker in the dark state, brighter in the bright state, and more of it gets to the membrane than earlier versions of glue sniffer. And this has really been a huge, huge improvement over the previous generation. So my lab has also developed a variety of computational tools for neuroscience. So this is including new optimization methods for doing deconvolution subject to different kinds of priors. It's methods for processing voltage imaging data, with which perhaps Ahmed will talk a little bit about, as well as tools for computational imaging. But lately we've been working on inferring how neurons convert input to output during imaging and optogenetics experiments where we might record glutamate and calcium and have some control over synaptic inputs. So we've been inspired by work from Tiago Branco's lab to sort of model individual neurons using customized little neural networks that reflect their structure. We can then use that glutamate and calcium imaging data to fit the parameters of that network to make predictions and guide our experiments. And the parameters of the network are interpretable quantities, like the strengths of synapses, which I think is going to be a good framework for characterizing the computations that happen in dendrites. One of the biological questions that we've addressed been addressing is the specificity of long-range inputs into cortex. So do neurons with a particular tuning in the thalamus, for example, project specifically to neurons with a similar tuning in the cortex? And if so, how does that wiring get set up? So JJ in my lab got at this question by optogenetically tagging thalamic synapses onto cortical neurons while doing high-speed glutamate imaging in cortex. And to validate that approach, he's done some really cool 4-color expansion microscopy to look for all of the components of the thalamic synapse. You can check out his poster on this at this year's SFM. So we think that all these different projects, we think of them as building an observatory for neuronal computation. So we want to be able to go up to a neuron and find out as much as possible about what its outputs are, and how it performs those transformations. This has been a very exciting goal for me to work towards. And if you're interested in working on this too, or if you know someone who is, please contact me. Thanks a lot for listening, and thank you to everyone who contributed to the work that I presented.
Yeah, thank you so much, Kaspar. That was really fantastic. Are there any questions? I don't see anything showing up in the group chat at the moment. So go ahead and type in your questions and we'll raise them. I had a question for you. You mentioned it piqued my interest using SLAP2 for optogenetic stimulation.
Yeah.
And that struck me as particularly challenging given the fact that, normally a single diffraction limited point is insufficient to drive, right? So if you don't mind, how are you getting around that issue of activating enough cell?
Let me see if I can advance the slides a bit. Okay, so that's right. A single diffraction limited point is not enough to drive most opsins, and so what you want to do is you want to illuminate an area, and so there's multiple ways to do that. One way is to create a diffraction limited point and then sort of scan it around in order to get more of the volume of the neuron, and that's pretty slow because if you have one of these diffraction limited points, you have to jump to every neuron that you want to image. It takes a few hundred microseconds to make the jump. At a kilohertz, you can really only do maybe 2 neurons at most. And the other way to do it is with two-photon holography. And one of the big problems there is speckle. So you only have control over the phase of a holographic pattern. And that means that, and actually, even if you had control over the amplitude as well, there are limits to what sorts of patterns that you can create in 3D. And so there are undesired illumination artifacts, and especially if you want to illuminate a low frequency area, like a patch that's large, then you lose axial resolution. And so that's been addressed with temporal focusing, for example. But basically, because the light is coherent, when you illuminate an extended pattern, then you end up creating sort of undesired artifacts outside of the plane that you, outside of the specific spots that you want to illuminate. So SLAP2 actually is it's sort of similar to temporal focusing. So temporal focusing actually effectively acts as a line scanning over a region that you want to image. And what SLAP2 does is it takes a line and it scans it across an open area of the DMD. So in this case, if we want to say image a bunch of rectangular regions of interest, then we would open these little horizontal slots on the DMD, and for every position there, we would scan the line horizontally across it at something like 20 kilohertz. But we can just open a pattern that corresponds to whatever it is that we want to stimulate, and then if we scan that line across it, then from the perspective of optogenetic stimulation, we're stimulating multiple pixels, so we don't have to worry about the saturation of opsin in a single point. But we don't have to worry about speckle because these things are on one axis illuminated sequentially, and on the other axis, there's a very low numerical aperture. I hope that explains a little bit to your
yeah, that was very clear. That's great. There was another question in the audience from Ben. Ben, do you want to unmute yourself and ask? Okay, so the question is, how do you deal with motion during imaging? Z motion in particular, since you're focusing on very small structures and need multiple scans to form an image.
Yeah, absolutely. So this is uh, so we published that SLAP the SLAP microscope, and there's a very extensive supplement on motion correction there. But essentially, we open up an area on the DMD so that horizontal motion doesn't affect us, because the amplitude of horizontal motion is smaller than the buffer area that we open on the DMD, and so we can align it out post-hoc. Now, vertical motion is a problem for any 2D imaging method. And the way that we deal with that is regardless, we have a 3D reference image that we take when we do SLAP imaging. And so when we're doing registration, we actually de facto do it in 3 dimensions. We basically find the maximum likelihood X, Y, and Z displacement for the image that we produce. We need to do this in order to have an accurate imaging model. What that means is that we know at least the position of the sample very, very precisely, to something like less than 100 nanometers on every frame. You can still lose data, or an object that you're trying to image. If it moves out of plane, it gets dimmer. We know how much we expect it to get dimmer by because we have a model of the point spread function, the microscope's function, and that's very important for estimating its brightness as the sample moves. So this is an advantage over other microscopes that don't have a model sort of behind the scenes. But of course, if it moves a lot, then you're not imaging it and there's nothing you can do.
Perfect. Okay, so I think in the interest of time, we'll move on to the next speaker. Thank you so much. Please join me in thanking Kaspar for a really, really great talk.
Thanks everyone for listening.
So our next speaker is Ahmed Abdelfattah. Ahmed is currently a postdoctoral fellow at Janelia Research Campus, and he's working on chemogenetic probes for imaging brain activity, amongst those certainly the famous Voltron and Campari. I met Ahmed when he was a graduate student in the lab of Robert Campbell at the University of Alberta, Edmonton, where he was really doing a lot of work pushing existing sensors more into the red range. Some really important work. And he was, at that time, he held a Vanier scholarship as a grad student, which is, for those of you not in Canada, our basically most prestigious graduate student award. He'll be starting his own lab at Brown University in 2021 at the Carney Institute for Brain Science, where he'll continue developing molecular tools for neuroscience research. And so with that, I'll hand the podium over to Ahmed.
Great. Thanks a lot, Ed, for the introduction. And thanks a lot, Kaspar, for introducing my talk very easy for me. The need for fast imaging of the brain is important. And I'll focus today on talking to you about developing voltage sensors to record brain activity at cellular resolution with millisecond time precision. Before I start, everything I talk about today and I don't talk about today, everything I do in the lab is available openly to anyone, or if you just ask through the email. So you can find the plasmids and viruses on Adgene, transgenic Drosophila stocks in Bloomington, and transgenic zebrafish lines at Zurich. And if you don't find anything there, just email me. All right, so what are membrane potentials and how do cells produce and maintain an electric potential? So all living cells have an inside and outside of, and these are separated by a lipid bilayer, electric insulator, if you may, and whereas media on both sides are aqueous conductors and ions can move around. You can have an imbalance in the charge across the membrane, and this leads to a voltage across the membrane. Every cell, every organelle, every living, every lipid-bound structure can support a voltage across its surface. And this voltage can change as these ions move back and forth across the membrane. For example, a typical electric field in a neuron in our brain is about 30 megavolts per meter. This is compared to a typical electric field in a lightning bolt, which is 3 megavolts per meter. So these electric potentials are huge, but they are localized to very small areas in our brain. So how do we go about imaging membrane potential in the brain? Really, every thought and action and memory is encoded by membrane potentials, like action potentials that are about 1 millisecond wide and 1/10 of a volt high. One way to go about imaging those membrane potentials is by impaling an electrode into a cell, just like I did here in the lab, for example. While it's very useful, you have exquisite temporal resolution. You can only record from one cell, one neuron at a time. Typically, the cell dies after you record from it, and it's very laborious and time-consuming. Ideally, we want some contrast agent, something to convert the voltage in the cell to an optical signal that we can see. And the challenges here is to find that something we can put in the membrane of a neuron, that it produces light, and it can react quickly and sensitively to the membrane potential changes that are very fast in time. And ideally, it doesn't kill your cell. And also ideally, we can target it to particular cells so that you can see what each class of cells is doing at a particular time. And remember, the signals we're after are those action potentials. So there are some challenges to image membrane voltage. And just to summarize maybe into three categories, the membrane volume is about 0.1% of the cell volume, and this leads to limited photon budget, and really trying to get brighter and more photostable sensors or probes would help a lot in getting enough signal to noise ratio to detect the membrane volume from a contrast agent. The second thing is you need to image fast to follow those fast voltage changes, and this limits the exposure time, which again limits the photon budget, and it puts constraints on the indicator speed. And the sensitivity and kinetics and the speed of the voltage indicators needs to be fast and sensitive enough to track action potentials, specifically for in vivo applications. So our approach to go about addressing those challenges is to engineer voltage sensors that are bright, photostable, and can be genetically targeted to a specific to the cells of interest. So the first, I'll talk about a couple of stories today, and the first story I'll talk about starts here. This is a photo taken from an airplane of the San Francisco Bay. This is the ocean here, and these are salt evaporation ponds. And the reason those salt evaporation ponds are purple in color is that these ponds have microorganisms living in them that are expressing proteins that absorb light. And these proteins are called microbial opsins. They are related in structure, at least, to the proteins to the rhodopsin proteins in our eyes that let us see. One of these microorganisms is Acetobularia algae. It's purple colored, again, because it's absorbing sunlight, and it uses this sunlight to pump protons through this microbial opsin outside of the cell to generate a proton motive gradient. So it's a form of primitive photosynthesis, if you will. We're interested in this protein not because it's pumping protons outside the cell. Others have used it as a silencer, for example, as an optogenetic tool. But we're interested in it because it absorbs light. It can change its absorption depending on the protonation of this retinal-bound chromophore. So the proton moves here, and then it protonates this chromophore, and this changes it. This molecule changes its absorption when it's protonated and deprotonated. We and others have shown that we can introduce point mutations to these proteins, where we can block this proton export outside the cell, and the proton sort of can move from inside the cell, protonate the retinal shift base, and move out again with membrane depolarization and hyperpolarization. And in this way, you can control the absorption of the retinal chromophore and the opsin absorption as a whole. And now if you add a fluorescent moiety here in close proximity to the opsin, you can control its fluorescence through energy transfer. So if it's close enough, the emission fluorescence from this fluorophore will decrease when the membrane is depolarized, because the absorption of the opsin increases when the membrane is depolarized. And this process is reversible. All right, so our way of going about making bright and photostable voltage sensors is by using this microbial opsin domain as a membrane voltage sensor. We linked a self-labeling tag, a halo tag, that can specifically bind small molecule dyes that have this anchor on them, the halo tag ligand, which is a chloroalkane. We were lucky in Janelia here. We were neighbors with Luke Lavis's lab, who makes some of the brightest fluorophores that are useful for live cell imaging. So we put those molecules on a chloroalkane ligand, and we thought, okay, so maybe we have the voltage sensing domain in the form of a protein. It can insert in the membrane, and we can capture this dye, specifically label this protein that's expressing in the cell, and we can end up with something like that, where the dye is only bound to the cells that are expressing this protein, and hopefully it can change its fluorescence with membrane depolarization. So when the membrane depolarizes, the dye would get dimmer and vice versa. Because this sensor platform is sort of modular, you can use different opsins. You can use different self-labeling tags if you want to do orthogonal labeling. You can use different color dyes. We call this sensor Voltron. All right, so this works in this works as a cartoon on a presentation. Does it work in real life? Here I'm just showing you a movie in cell culture. Here I'm patched on this neuron, and we're recording it both with an electrode and Voltron, monitoring Voltron fluorescence after labeling the cells with the dye. As you can see, the fluorescence from Voltron can track individual action potentials and subthreshold changes. This movie is slowed down several fold. This whole trace is about 1.5 seconds long. All right, so this works with different color dyes, as I mentioned before. And depending on the dye color you add, you can record membrane potential in from green to far red. So this works well in cell culture. You can easily add the dye in cell culture, but there's an extra step of needing to add a dye. And what I hope to show you in the next several slides is that it's useful for imaging membrane voltage in vivo too. And this extra step of needing to add the dye is useful and well worth it if you're interested in precise correlation of activity in vivo. So the first model organism I'm going to show you that we used Voltron in is zebrafish. This is a transgenic zebrafish line where most neurons are expressing Voltron. This is just a Z-stack from top to bottom. The eyes of the fish would be here and the tail would be down here. The way we label, and this is another one with the localization to only the soma of the neurons. And the way we label the fishes, we let the fish swim in water with dye spiked in it for a couple of hours, and then we put it in clean water for an hour, and then you're ready to image. So introducing the dye is very simple in this case. So we collaborated with Misha Aron's lab here at Janelia, and there we used a light sheet microscope to image fish during fictive behavior of paralyzed fish in a closed loop virtual reality system, where we display a moving grating to the fish, which induces swimming of the fish, and we can record the swimming using electrodes. Here we're showing Voltron that's expressed in the midbrain nucleus, and we can record from now about 10 neurons over about 10 minutes simultaneously. And if we just zoom in to this small square here, you can appreciate that we can correlate the neuron activity at single action potentials with the behavior of the tail, behavior of the fish, which is very fast in the order of milliseconds. This whole trace here that's zoomed in is about one second. So if you would do something like calcium imaging, you would see a big bump. Whereas here you can see individual action potentials, and you can correlate it with the behavior you're seeing. Using this, we classified these neurons based on how their subthreshold and spiking activity changes around the start of a swim bout. That's represented by this dashed line here. This is just an example showing each class of neurons where one of the classes sort of turns off and shuts down once this fish starts swimming. Another one ramps up its subthreshold activity and starts spiking before this fish starts swimming. And the other one sort of starts spiking and starts being active after the fish starts swimming. And we can classify many neurons over many fish using that method, and we can sort of build spatial organization maps. And we can start to build models where we can classify the different subsets of neurons and correlate that with a sort of a motor circuit where the one class of neurons is inhibiting, the motor circuit inhibits its activity, another one sort of triggers the motor circuit, and then some sort of late internal copy of the activity sort of for the fish to remember where it moved and sort of base its behavior on that. So just a quick note here where, you know, we imaged, in this case, 179 neurons at the time. Now we have many more neurons. But to do something like that, you would need to do 179 separate experiments and go in to do whole cell patch clamp recordings of 179 neurons in this particular nucleus in the brain. Here we imaged it over 10 fish, 10 cells at a time. So it sort of makes those experiments significantly easier to do in the lab. The other model organism I wanted to show where we used Voltron is Drosophila fruit flies. And here we're expressing Voltron in a couple of dopaminergic neurons in the mushroom body. What Glenn Turner's lab here and his postdoc Yuqin Shui are interested in is navigation and memory in Drosophila. So what I'm going to show you here, this is a movie of a Voltron expressing fly that will navigate towards an odor, and we can record spiking and electrical activity on in the neuron live as the neuron as the fly is behaving, and we can do this for tens of minutes. And this is important for Glenn's lab and for Yichun, because they're interested in memory behavior, in memory formation, and their behavior tasks takes tens of minutes. And compared to sort of similar voltage indicators that rely on something like fluorescent proteins, for example, which don't have the added complexity of adding an extra dye and would work very well if you're interested in recording maybe for 1 or 2 minutes, Voltron can record up to 10 times longer compared to the same opsin bound to a bright green fluorescent protein like amnion green, for example. Yeah, and this keeps going on. And you can introduce, in this case, the dye to the fly either in the food or by bathing the brain in the dye for a couple of hours before you perfusing the brain in a dye solution before you start your imaging. All right, the third model organism I want to show you is maybe more relevant to many of you in the audience today, is the mouse. And this is just a cranial window on top of the visual cortex of a mouse that's expressing Voltron, soma-localized Voltron. The dark lines here are the blood vessels, and the single spots here are actually single cells labeled with Voltron. And the way we introduce the dye here is by injecting the dye in the bloodstream. And it can cross the blood-brain barrier and can find the cells only expressing Voltron and does not label the other cells. And that's why you see, you can get this contrast and you can see those bright spots. If you zoom in, these are sort of single neurons labeled with Voltron, and the other cells that are not expressing Voltron you can't see. All right. This is, again, an image of the cranial window over the primary visual cortex. And this is just an example of a field of view that we can image from using a standard wide field fluorescence microscope. Here we're imaging from layer 1 interneurons. And in this field of view, for example, there are 40 cells segmented. And these are the fluorescence traces from each. we can zoom in into these three areas of these traces and assign individual spikes and track sub-threshold voltage changes. And this shows that we can extend voltage imaging from a couple of neurons for a couple of minutes to imaging like dozens of neurons for tens of minutes. And imagine the difference between having a 40 factorial and 2 factorial order of magnitude and changes or sort of correlation of activity of the different neurons in the brain. All right, now I want to switch gears a little bit about talking to you about ways we're trying to improve the signal detection from voltage sensors in vivo. All the fluorescence traces I've showed you so far and all the fluorescence traces of most voltage sensors actually that are used right now in vivo are negative going, meaning that when the membrane potential increases, when the neuron spikes, the fluorescence from the voltage indicator decreases. So it gets dimmer. And so to improve SNR, especially in scattering tissue and dense labeling in vivo, we need to sort of have positive going sensors. And this is just a simulation based on the imaging data we did in mice from Voltron that we did in collaboration with Kaspar to sort of reiterate that point where, this is just simulated positive going data based on negative going data from Voltron. And the white line here is sort of the error rate in detection of an action potential, and it falls more rapidly if the sensor is positive going. So if you improve the sensor dynamic range of a positive going sensor, you will improve the error rate faster than a negative going sensor. And it's very clear in a more dense sample, for example, the error rate increases dramatically from a negative going sensor because your baseline fluorescence is bright, and that contributes to the signal, the photons you're imaging. But with a positive going sensor, which is dark at rest, you can image from a much denser sample. So we went back to the drawing board and looked at this protein in more detail. So here I labeled, I'll go into a little bit of protein engineering here mainly because I think it's cool, but it's also important for developing the sensor. But if I go back to this schematic, for simplicity, there are three positions that are involved in this transporting this proton from inside the cell to outside the cell in this algae. Here, these are negatively charged residues that will accept a positively charged proton, and these are labeled proton donor, proton acceptor, and proton release. So it's sort of, you can imagine the proton is hopping between those three different amino acids to reach the outside. So we thought, okay, for Voltron, we mutated that position and blocked the proton from going outside, and it can only reach the retinal chromophore here. But that led to a protonation from the inside of the cell when the membrane depolarized, and that led to a fluorescent signal that goes down when the membrane depolarizes. So we thought, okay, so what would happen if we block the proton from the outside? If we block the proton from the inside, can this protein accept protons from the outside? And sure enough, we can. If the membrane depolarizes, this specific variant can increase its brightness when the membrane depolarizes. But I successfully made a crappy voltage sensor that's not fast enough to detect single action potentials anymore. But the signal is in the right direction. It increases when the membrane depolarizes. So we thought, okay, let's improve the access of protons from the outside. So we mutated that position into a negatively charged residue such that protons can reach the retinal chromophore from the outside easier. And sure enough, we can restore the kinetics of the sensor. However, the dynamic range of this sensor is much smaller than Voltron. So we mutated the last position that was responsible for the proton release, and now we restored the dynamic range and sort of made a mirror image of Voltron, where when the membrane potential increases instead of decreasing with Voltron, with 3 rational point mutations, we can increase the membrane potential. We can increase the fluorescence with an increase in membrane potential. And because it's the inverse of Voltron, we call that sensor positron. And the real cool thing about this, protein engineering effort is that it works with all opsins. So not only the rhodopsin that's used in Voltron, but all other opsins. We tried it with MAC rhodopsin, for example, or ACE1 rhodopsin, and we show other examples in the paper now that just came out a couple of weeks ago. But it switches all those opsins into positive going sensors. And I think this is a sort of a good platform to sort of build on and generate the next generation of voltage sensors. So I hope I've shown you today, I think I'm running out of time, but I hope I've shown you today sort of the, with the limited membrane volume we have, the limited photon budget, the chemigenetic approach of adding the extra step of adding a dye helps improve that. And if it's important for you to image for more than a couple of minutes and more than a couple of neurons, it might be worth it to use. Voltron and positron both can track action potentials, and it's they can be used for fast acquisition of single action potentials, and sort of positron paves the way for improved positive going voltage sensors in the future. A shameless plug, like Ed mentioned, I'm starting my lab at Brown next year, and we're hiring. We're going to continue making molecular tools for brain science, and if you're interested in joining the team, please reach out to me. Science is a team sport, and I would have not been able to do any of this without beautiful collaborations like with Kaspar, for example, who's here, and many others at Janelia and everywhere else. I'll just single out a couple of people here. Eric, who's who I did my postdoc, who I'm doing with my postdoc with in the lab here at Janelia. Luke Lever's lab, who made the bright fluorophores. Misha, who made the all the in vivo work we made with Misha and Neuro, all the in vivo work in fish. Carl's lab, Carl's Ruthazer lab for the mouse imaging, and Glenn Turner's lab for the fly imaging. And with that, I thank everyone for your attention.
Thanks, Ahmed. That was fantastic.
Thank you.
There is a question already from the audience, and maybe I'll ask it straightforward. There was a question about how long you were able to record the Voltron signal in cell culture and in mouse cortex, in the mouse cortex.
So we were able to record for a couple of like 15 to 20 minutes in cell culture or in the mouse cortex, and we show examples of that somewhere in the supplement in the Voltron paper. And we can routinely record up to 10 minutes in any preparation. It really depends on how deep your cells are, how much light you're exposing your sample at, but I would say we can safely record for 10 minutes or more.
That's great. Another question from Ben. Does having the Voltron and tethered dye at the membrane change the lipid environment and affect the kinetics of other native ion channels?
Inserting anything in the membrane will affect the membrane. We show that having the dye is no different than having a fluorescent protein there. So the dye part doesn't make a difference. Of course, if you overexpress any membrane protein, it will have adverse effects on your neuron. We show that it doesn't, at least in cell culture, we did patch clamp experiments to record series resistance and capacitance of membranes, and there was no significant difference we can see in cell culture. So I wouldn't say that the dye adds any issue there.
Okay, great. Camila, did you want to ask your question, or should I ask? You can unmute yourself if you'd like.
Sorry, I can't see I'm trying to get the chat, but I can't.
Yeah,
see
I think it's hard
when I'm sharing the slide. But
I'll ask Camila's question, which is, first of all, she said it's an awesome talk, which I agree with. Can you detect changes in voltage at dendritic and spine levels?
We're working on that. We're actually working on that very closely with Kaspar now. It's in the works. So far, what we have been able to reliably do is at the soma, but this work is in progress, and we're basically, the way we're addressing that is by trying to improve the dynamic range of the sensor and the speed of the sensor and better localization at synapses. So this work is in progress. It's probably the first versions that I showed today won't do that very well, but it's in the works.
Well, along those lines, I was wondering, I mean, one of the things that gets released along with glutamate at a synaptic, from a synaptic vesicle, I think are protons. And so will the very local change in pH affect the reporter in some nonlinear way?
So I did some experiments where we tethered the fluorophore to the surface of the cell without the voltage sensing domain part, and it didn't seem to have a huge any change in fluorescence. And really, those dyes in particular have a pKa that's shifted to the left, so it would require significant acidification of the environment to change the fluorescence of those dyes.
So another question, Ankit, do you want to ask, or should I ask?
Sure, I can go ahead. I was just wondering how the phototoxicity is with these voltage imaging experiments, because if I'm right, that was one of the major issues holding them back, that you need very high light intensities to image. Sorry if I missed this information.
Yeah, so that's a good point. With those dyes, because they're bright and photostable, you don't need that much light. In fact, we need an order of magnitude less light to image the dyes compared to fluorescent proteins. So if you have the same sensor with a fluorescent protein in it or a dye, you need an order of magnitude less light to image the dye one compared to fluorescent protein. So we have shown in vivo that we can record for tens of minutes, and we didn't see any adverse effects on the cell. In fact, we show one example where we image the same cell over a period of a couple of months, and we return to the same cell every couple of weeks and image it again and again for several minutes, and it still behaves similarly.
Thanks. There's a question from Ronan. Ronan, did you want to ask, or?
Yeah, sure. Hi, great talk. I was wondering, when you use Voltron in mice, you said that you deliver the fluorophore by injection into the bloodstream. I was curious to know how long after the injection you start to see the fluorescence in the brain, and then after that, how long does it last? Does it last for a few days, or? Yeah,
that's a good question. So we start imaging 24 to 48 hours after injection of the dye, so to give some time for the dye to clear out of the brain and to deliver it to the cells. And then once the dye is bound, it's covalently bound to the protein. So it depends on the protein turnover. So we have imaged between 24 hours after injecting the dye up to 7 days after injecting the dye, and we can still see signal. If you want to image after 7 days, we would inject another batch of the dye and just label the newly synthesized protein. But it's really a matter of how fast the protein is turned over depending on the cell you're imaging at that point. But we give it 24 hours at the beginning, sort of to label the protein and to clear the dye, the extra dye that's unbound.
And there's one last question from Simon. Do you want to ask?
Yeah, I can do that. Thanks for the talk. It was great. I was wondering that due to the fact that Voltron is based on an opsin, and this opsins conducts photon, I'm just wondering if there are any currents that are evoked by the light used for imaging when you image Voltron.
Yeah, so the quick answer is no, and I think I have a slide on that here. So this is the photocurrent from the rhodopsin without any point mutations to it. So you can see there's some inward current, outward current when you shine light. So this is just shining light for a second when the cell is expressing this protein. And then with Voltron, you have a transient current at the beginning, but no steady state current. So you can imagine if you're recording for tens of minutes, you'll have a transient current for the first couple of milliseconds, and then you have no current And the same thing with, for example, positron. You have a transient current in the opposite direction because we reversed the proton transport chain, but you have no steady state current. For the imaging experiments, you have a transient current with all the opsin based voltage sensors out there. You have a transient current in the first 1 to 2 milliseconds of imaging, but afterwards, once the light is on, you don't have any current, steady state current, because the protons are not being actively pumped out. It's really, this transient current is just the residual protons at the retinal moving in or out of the cell at the first couple of milliseconds.
Great. Okay, so I think what I'll do is just bring the formal session to a close. I'm going to stop your sharing and start just a slide showing our talks. Oops, wrong talk, sorry. Talks coming up. I don't know why that showed up. Okay, I think you can see it now. So first of all, please join me in thanking both our speakers, Ahmed and Kaspar. It was a really fantastic set of talks, and I think it was great to have this thematic connection. I think the two of you are solving very similar sorts of problems, and the synergy of your techniques is going to be amazing. So thanks. And just a plug for our next and last session. So next week, we'll have talks by Mihaela Yordanova from Concordia University. She'll be talking about VTA dopamine transients regulate temporal difference learning, and talk from Alex Lohman, who's in Calgary, and he'll be talking about novel optogenetic tools to investigate Pannexin-1 signaling in the CNS. So please join us next week as well. And if you'd like to stay around now and ask questions of our speakers, if they're willing to stay on and chat, we can open the floor and just have like an informal discussion. So.
Hey, Ahmed.
Hey, Kaspar.
I have a question.
Sure.
So the photocurrent got me thinking. I mean, I know this isn't the main reason why Voltron doesn't work under two-photon, why traditional Voltron doesn't work under two-photon illumination, but transient illumination would repeatedly elicit, there's some like molecular capacitance essentially, right? I mean, there's just capacitance of the indicator. And so transient illumination might actually be fundamentally different than continuous illumination from that perspective. I mean, obviously you can tell the transient is much slower, and we've done continuous two-photon illumination. There's all these things, but do you think that that's a contribution to the
yes, yeah, I definitely thought about that, and I did the same experiments in using your scope basically, and the time scales of the transient current at the beginning is longer than the transient illumination on your scope, at least, or on a raster scan. So it's not an issue. It's to the protein. It's still continuous illumination, basically.
Yeah.
So it's not an issue. But that's a good point. That's what when I was doing those experiments, that's the first thing I thought about. It's so nice to see some very good old friends on the talk here. So.
Yeah.
It's a great group of people. I hope that everybody's going to be willing to chime in if they have questions or comments. There's a lot of comments on the chat. You might want to take a look, which are mostly basically how great your talks were. So, I guess Ahmed, you're always going to be an official Canadian in my mind. Although I guess now you're really.
I treasure my time in Canada, and I would like to think of myself as partly Canadian at least.
We'll let you.
You should give a plug for your lab. What are you going to focus on?
We're going to focus on making, in general, molecular tools for neuroscience. So sort of continue working on voltage sensors, other types of sensors, probably neuropeptide sensors, and some really cool ways of genetically tracing and manipulating neurons in the brain, sort of whole brain circuits. So if anyone is interested, please get in touch. We're hiring. Both me and Kaspar are hiring. So yeah.
Neuropeptide indicators via the GPCR loop 3 strategy or something different?
That's the one of the strategies, but not the main strategy. So via sort of phage display and nanobody and antibody engineering, probably.
Cool.
So different ways of going about it. We'll try several things and see what works best.
Yeah, I'm always looking for a D-serine reporter. Anytime you can report D amino acids, I'll be signing up.
Cool.
I mean, there is some D-serine response in the in the glycine indicator. Ed, we should talk offline more about neurotransmitters.
Yeah, no, that would be fun. That would be cool. Okay. If there isn't any burning questions, you guys both gave a fantastic talk, very clear. So thanks again. And I guess we'll close the session. Make sure to join us next week. I really appreciate you guys taking the time to give these talks. They were really good.
Thanks for
Ed, for having us.
Bye-bye. Good luck with your labs.